Hi, everybody. My name is Will Swain. I am a first year, or rather first semester PhD student here at University of Wyoming. And my sort of research topic over the next few years is going to be about population genetics of elk and chronic wasting disease in the state of Wyoming. So for my GIS project, I wanted to use some of the data that I've received so far and ask the question, can landscape variables help predict where to find chronic wasting disease positive elk in Wyoming? So for a little bit of background, chronic wasting disease is a type of transmissible spongiform encephalopathy, or TSE. These diseases are caused by a prion, which is an infectious protein particle that essentially gets into a host and causes continued misfolding of similar proteins, which can lead to fatal neurodegeneration in infected hosts. So chronic wasting disease specifically was initially identified in a captive mule deer just over 50 years ago, but it is now known to affect many different types of cervids, including, but not limited to, elk, pronghorn, and moose. So these prions causing chronic wasting disease are known to be environmentally persistent, which is one of the challenges about understanding the transmission and risk of the disease to wildlife. And it's shed in animal excretions and in tissues from carcasses, for example. The current distribution of chronic wasting disease in both wild and captive cervids encompasses 26 US states, 3 Canadian provinces, South Korea, captive populations of cervids there, and Scandinavian nations, so Finland, Norway, and Sweden. And so just a little bit about elk. As of 2018, there were over 100,000 elk in the state, according to Wyoming Fish and Game. Obviously, looking at their historical range, their numbers and distribution are much reduced, but overall, they're considered a pretty healthy and robust species, both in Wyoming and throughout the sort of western states. So their home range in Wyoming is primarily along higher elevations in the mountains, so that basically means western portion of the state, especially near Jackson and Yellowstone. In the northwest area of the state is the National Elk Refuge and Feed Grounds, which is an interesting and very controversial topic that I'm not going to get into here, but that's where a lot of elk, including the Jackson Hole herd, exist, and also the southern and sort of southeast portion of the state, so where the Southern Rockies and Laramie Mountains are. They are a highly social species forming herds. They tend to migrate to the higher elevations in the summer for grazing and in late summer for breeding purposes. And then when the harsher winter weather and climate conditions descend on the state and area, they like to migrate to lower grassland areas for browsing and feeding. So the objectives for my project were primarily to plot locations of known chronic wasting disease positive elk that were submitted to Wyoming Game and Fish from 2000 to 2015, or sorry, 2011 to 2015. And I wanted to do this as much as possible in ArcPro. I'm very much a novice using ArcPro and R as well. So all things being equal, I decided to try and challenge myself to use this particular software that I may not get more opportunities to do in other classes or realistically in a lot of other research down the road. So I wanted to try and use ArcPro as much as possible. So using this data set that I had, I planned to set a study area, and these elk data points were collected from hunters during hunting season, so that is to say early fall to early winter in the state. Then I wanted to apply landscape and environmental variable overlays to the study area, and then determine any possible associations between these landscape variables and the presence of chronic wasting disease confirmed elk. And then use that information to try and predict where we might see chronic wasting disease in elk in the future. So my study area, initially I was planning on doing the entire state of Wyoming, but as you can see from the data points on this map, they're pretty much all clustered in the southeast corner of the state. So it really made sense for me to focus locally as opposed to do the entire state for this particular analysis. So here is a map showing sort of the extent of the study area that I set somewhat arbitrarily, where it exists in the southeast corner of the state. And I did a nice little color coding thing to show which elk coordinates correspond to which year they were submitted and confirmed to be chronic wasting disease positive. So even though they're separated out here, for the purposes of the study down the road, I just treated everything as one lumped data set. So I didn't do any temporal analysis or split them by year for anything other than sort of data visualization purposes. So my data inputs, I had 81 chronic wasting disease positive elk reported with accurate and sufficient coordinate data that I could include in this analysis. So the breakdown is there for when they existed and how many. And like I said before, group them together for subsequent analyses. So my landscape variable selection, I was primarily interested in understanding what sort of influence land cover type may have. So that led me to download the National Land Cover Database raster. And I reclassed and simplified it to 6 primary cover types as opposed to the full spectrum. So water, developed land, barren land, forest, sort of a grassland, shrubland mix, and cropland. Then I obtained a 30 meter resolution DEM from the ArcPro Living Atlas, set to my region of interest, rescaled to 90 meters, and then I used this raster to calculate 2 other geomorphometric rasters. So the Compound Topographic Index, or CTI, which is simplified and in essence a wetness index, or sort of wetness potential index, and then the surface area ratio, which is in essence a steepness index. So I also wanted to do a heat load index using Jeff Evans' really handy toolbox. I could not get it to work for my data and in ArcPro, unfortunately. So I kind of had to scrap that, something I want to pick up in the future though. So here's a brief overview of my raster layers. On the sort of top left, we've got the land cover raster and the 6 reclassed and simplified land cover types that I use in my report. We have the 90 meter DEM elevation raster. So the white areas obviously represent higher values and greater elevation. And we've got the surface area ratio raster. So pink areas represent higher values, which mean greater steepness. And lastly, the CTI raster. So blue areas represent higher moisture potential. So then I wanted to think about distance rasters. So, proximity to certain either land cover types or, from the chronic wasting disease positive elk. But in order to effectively analyze this later as sort of a binary outcome, presence or absence of chronic wasting disease, I needed to populate my map with random points, which represented pseudo absences. So I used a data point, random data point generator of 10 times my sample size. So I had 81, so I added 800. And I retained the colored buffers just for, like I said, visualization purposes to sort of better understand where my actual real data points of chronic wasting disease positive elk were compared to all the pseudo absent points generated on my map. So from there, I actually broke down the NLCD raster layer I already had into 6 individual land cover type rasters, and then I converted those into distance rasters for each of those 6 land cover types. And so those are presented here for the cropland, grassland, forest, barren land, developed land, and water distance rasters. So I used the same sort of color scaling scheme. I thought it was an easier way to visualize. So gray represents essentially 0 distance, or at least really close proximity, going up to red, then yellows, greens, blues, and purple pinks being farthest away. So I thought this was a really neat way of visualizing according to the data points, both real and generated, distance sort of understanding in my map. And then despite some initial reluctance, I ended up needing to use R for the statistical analysis. There actually is a generalized linear model function within ArcPro. I used it, could not get any real sensical data out of it though. So I think Shannon was vindicated there. So what I did was I extracted all the underlying raster values at the 881 data points. So the 6 distance rasters and the 3 geomorph rasters. And I also assigned binary values for either presence, so for my real data, or absence for my generated data points of chronic waste and disease. And then I exported that data, brought it into R, and performed a binomial linear model to determine any factors that might be correlated with the presence of chronic waste and disease. So to ensure that none of my predictor variables, of my 9 predictor variables, were autocorrelated, I performed a Pearson correlation. Nothing was above 0.7, so that all checked out. And then I selected the candidate variables from the linear model produced based on their significance measurements, and determined that 4 of my 9 variables seemed to indicate statistical significance as possible predictor variables. So those were forest distance, grassland distance, crop distance, and ground surface elevation, which is my elevation or DEM raster. So to ensure that the overall fit of the linear model I produced was reasonable, I used the receiver operating characteristic curve to check the overall fitness. Calculated area under the curve was 0.8399, which exceeds that sort of minimum threshold of usefulness of 0.7 or 0.8, depending on who you're talking to. So I knew that my overall model, despite only having less than half of the predictor variables seemingly useful, was an okay model overall. So then I took the coefficients of the candidate predictor variables and brought those back into ArcPro and did a little bit of raster algebra against the distance rasters and DEM raster to create sort of a new raster layer, which are the predictive.
Apologies for that. My microphone seems to have failed me, so I had to switch back to the computer. Anyway, as I was saying, so here are the 4 prediction raster layers. So the green areas represent areas of better predictive value, or in this case, increased sort of likelihood based on the predictor variable of finding chronic waste and disease positive elk. Whereas red areas, unsurprisingly, are the opposite. They are either poor predictors or just generally less likely to find chronic waste and disease positive elk there. So then once I had these 4 predictor rasters, I combined them into one unified prediction raster, which is sort of the ultimate goal of my project. And now my slides won't move. There we go. And here we go. This is the unified predictive raster layer for chronic waste and disease positive elk in Southeast Wyoming, which I thought was a pretty satisfying result. So in terms of sort of conclusions, questions, and future steps, it does seem to be that there is evidence that landscape and environmental variables may influence the presence of chronic waste and disease positive elk. It's unlikely that these variables are directly driving disease transmission, but maybe due to behavioral changes or local adaptation based on environmental landscape features, elk are behaving in different ways, which may facilitate or sort of hurt the ability of chronic waste and disease to spread throughout populations. There are known elevations or associations with elevation, obviously, with elk. They tend to range in higher altitudes for summer grazing, but tend to browse in grasslands during winter, which happened to be the time period of data collected for my project. So it maybe makes sense that grassland and scrubland features were really highly associated. I'm curious to find out in the future what influence forest areas have. I don't entirely know, and I couldn't really hazard a guess right now. Something I did find interesting is that other research suggests that chronic waste and disease infected animals frequently drink and urinate, and consequently seek water sources. So I was a little bit surprised that there was really no association with water, whether that be the moisture index or the actual land cover type for water found in this study. And I also thought the correlation with cropland and developed land to be interesting too. So, the question then becomes, maybe there are some behavioral changes in elk that are infected, brought about by neurodegeneration. Maybe they're less afraid of humans or encroaching on sort of human territory. So for future steps, I'd like to broaden the study area, obviously, look at the entire state of Wyoming. And I'd also like to bring in from my data set include known chronic wasting disease negative elk. I have thousands of those data points, and I've kind of felt like it would detract from the overall nice clustering of stuff going on right now. So I didn't incorporate them this go around, but in the future I definitely will. I'd like to look at more landscape and environmental variables, especially doing more geomorph analysis. Like I said, heat load index was, it's a bit of a no-go this time, but I think in the future it would be useful. And then of course, maybe a little bit less directly related to GIS work, but my overall research is going to focus largely on genomic research. So understanding population relatedness of elk throughout the state of Wyoming, genetics relating to chronic wasting disease itself, and then sort of trying to combine those two. So thank you very much. That was my project, and looking forward to any and all of your questions.